So I gave some thought. I was asked to just sort of give a overview of immunity to fungal pathogens, and I thought about talking about my research, and it's rather specialized. So rather than do that, I thought I would just give sort of the view from 30,000 feet. So for some of you, this may be mostly stuff or all stuff that you already know, but hopefully there'll be something in there that people will learn from. So my objectives here, this is the cone of learning, and you'll remember 90% of it if I do a dramatic presentation simulating the real experience. It's kind of hard to act out fungal immunity too much, so I'm not going to do that, and I'll be happy if you're in this 10% category here. But I'm going to give you a broad introduction of how fungi are uniquely recognized with an emphasis on the human immune system, although most of our knowledge is from the mouse, and also how specific disorders of fungal immunity are associated with specific mycoses. So before I go into this, this is a slide that Matt Fisher would be better at showing than or talking about than I would, but this is the famous tree of life. And for those of us who believe in evolution, here are the eukaryotes on the tree of life. And if so it's just basically a branch from the tree. And then if you look at fungi and humans, they're sort of smaller branches. So why am I showing this? Fungi, the point is that fungi are very similar to people, especially on the cellular level, and indeed, a lot of Nobel Prizes have been won by people who study basic cellular mechanisms in model yeast, such as Saccharomyces. There are, of course, some differences. Fungi have ergosterol as their main sterol, or most fungi do, rather than cholesterol. And of course, fungi have cell walls, and most of our antifungal drugs, the azole antifungal drugs, amphotericin, they use ergosterol as their target, and the echinocandins, of course, inhibit cell wall synthesis. So, but for the most part, one of the problems with antifungal development has been because of all these similarities. And following fungal invasion, the host innate immune system sees and responds primarily to the cell wall components, because that's what's on the surface. So, that's what the immune system sees. And fortunately for us, the immune system has adapted very, very effective defenses against fungi, and some of these are evolutionarily conserved. Here's the horseshoe crab, Limulus. It recognizes 1,3-beta-D-glucan and forms a cascade which winds up clotting the hemolymph, the sort of the blood of the horseshoe crab, and that leads to warding off the infection in this beast over here. And this, of course, is the basis of of assays for detecting beta-1,3-glucan, such as the Fungitel assay. Here's Drosophila, the fruit fly. It has a beta-glucan binding protein. When that's activated, it stimulates a signal cascade, which includes toll, and that leads to an antifungal response, and that, of course, shared the Nobel Prize. And humans here, we have Dectin-1 and other mechanisms to recognize beta-1 glucans. So there's sort of a battle of the kingdoms, so to speak, and for the most part, and all throughout history, fungi have either won, or you have somewhat of a stalemate where you have colonization or latency, but the fungi aren't really causing much in the way of problems. What's happened, though, in the past half century or so is that the fungi have started to win, and that's because we have huge populations of immunocompromised patients. So the fungi have sort of gone viral, if you can excuse the poor pun. And we've heard about the so-called 1.5 million people who die every year from fungal infections. They're somewhat pie-in-the-sky figures because, for the most part, fungi aren't reportable diseases, but I think they're reasonable estimates, and regardless of whether it's 1 million or 2 million or 3 million, it's a lot of people dying from fungal diseases. So you've seen the model cell wall. Danny, others have, Fiona have shown. This is Candida albicans. It has a manoprotein outer layer, and on the inner layer there's beta-1,3 and beta-1,6 glucan, and some cross-linkage with chitin. And the innate immune system is able to recognize all of these different components, as I'll get into in a minute. However, every fungus is different. So, and even among strains of fungi, there are important differences. And the host has to contend with all of these differences. For instance, my favorite fungus, Cryptococcus, it's got a capsule. So here's the cell wall. So we have the beta-glucans, the chitins, the mannans. They're over here, but then you got this huge capsule, and that masks the cell wall, and the immune system isn't able to see, for the most part, any of those components. Here's histoplasma, and some strains of histoplasma, not all strains of histoplasma, make alpha-glucans. Alpha-glucans don't seem to be well recognized by our innate immune system, and the alpha-glucans actually mask the beta-glucans. So each fungus presents unique challenges to the host, and fungi come in all sorts of sizes and shapes, what people like to call morphotypes. And many fungi change the size and shape. So Aspergillus, for instance, what we're exposed to are Candida, the spores in the air. We breathe thousands, sometimes millions or billions of them on a daily basis. So the host has a crack at killing the Candida. If that fails, then the Candida will germinate and form hyphae. The hyphae become very large. They're too large to be phagocytosed. But fortunately, we have very good defenses. We have neutrophils, we have macrophages, dendritic cells. They can gang up on the hyphae and kill or inhibit the organisms. There's also a category of fungi, the thermally dimorphic fungi, like histoplasma, blastomyces, coccidioides, and they undergo phase transition in the host. So out in the environment, they may be mostly in the form of molds, and then when they get into the host, they undergo phase transition and now the host has to contend with yeast forms. Routes of invasion. Fungi are attacking us, or we're being exposed to fungi from all sorts of surfaces. We're breathing into the lungs. So in the lungs, you have the alveolar macrophages, you have the epithelial cells that are very, very well adapted to take care of Candida and have a balanced response. You don't want too much inflammation. Then, of course, there's skin, mouth. We heard about the GI tract from Neil, sinuses. The host has to contend with all these fungi and figure out a way of containing it without having too much inflammation. However, what we've done with modern medicine is we've circumvented a lot of these responses. So we put IVs in our patients, and then the Candida from the skin is able to get directly into the bloodstream. We've had, at the hospital that I work at, we've had, in the last 7 years, we've actually had 24 cases of IV drug users who've had candidemia from presumably from IV drug injection. So they're basically bypassing the system, probably injecting Candida directly into their blood. We have Foley catheters, Ommaya reservoirs, et cetera. So we're doing things that, this may not be what you traditionally think of as immunocompromise, but we're facilitating entry of fungi into places where normally it isn't there. So how do we recognize fungi? Microbes have pathogen-associated molecular patterns, so-called PAMPs. Those are constitutive and conserved products which are not expressed by animals, and these are seen among bacteria, viruses, fungi, parasites, et cetera. And innate immune recognition is largely based, so when you say innate, you're talking about germline-encoded receptors, and the innate recognition is largely based on receptors recognizing these PAMPs. There's 4 major classes of pattern recognition receptors. The most important class in terms of recognizing fungi is probably the C-type lectin receptors, which you heard about a little bit from Darius. There's also toll-like receptors, which Darius mentioned. There's nod-like receptors, which are important in inflammasome activation. And there's also Rig-I receptors, which seem to be more important for viral recognition, and people haven't shown much of a role yet, at least as far as I know, in terms of fungal recognition. So I'm going to talk a little bit more about these C-type lectin receptors. And they're a class of receptors. They mostly bind sugars, specific glycans, using conserved carbohydrate recognition domains. I say mostly sugars. For those of you who've seen the latest issue of Nature, Gordon Brown has a very nice paper showing a C-type lectin receptor that actually recognizes a component of melanin in fungal Candida. So these C-type lectin receptors are extremely important for recognizing fungi, and they, as I mentioned, they recognize specific components. So Dectin-1 is specific for beta-glucans, Dectin-2, 3, Minkle, mannose receptors. They recognize different components of mannans on the fungal cell wall. And importantly, recognition is followed by signal transduction events. So you have, the sick, and that leads to pathways that include CARD9, and that in turn leads to functional responses, which allows the host to control or eliminate the fungal pathogen. And a lot of these responses, I'm sure you're quite familiar with. Now, multiple innate receptors are stimulated by fungi. So a lot of times when we study fungi or pathways, we tend to use a reductionist approach, and we study just one receptor or one ligand. But in actuality, when the innate immune system sees a fungus, it recognizes multiple ligands on the fungus. So for Candida albicans, mannans, they're recognized by various different mannose receptors, several different ones, in fact. They're also recognized by TLR2 and TLR4, two toll-like receptors. The beta-glucans on Candida, although they're somewhat masked, the ones that are exposed are recognized by Lectin-1 and other beta-glucan receptors. Beta-glucans are very potent activators of the complement system, and that leads to complement deposition on the organism, which leads to recognition by complement receptors, of which there's at least 4 different ones on human white blood cells. There's also natural antibodies against beta-glucans, and then there's acquired antibodies too. So all of these factors contribute to recognition of just one fungus. And each ligand receptor system, and this is a concept that's been advanced very nicely by Mihai Netier, each ligand receptor system activates specific signaling pathways. Some pathways are actually quite pro-inflammatory, whereas other pathways seem to be more involved with putting the brakes on the immune system. And the immune system, the immune response to the fungus, at least the innate immune response, is determined to a large extent by how much each of these different pathways are stimulated. And then, of course, innate immunity winds up linking to acquired immunity. And so when you have stimulation of the C-type lectin receptors, toll-like receptors, that instructs antibody and T-cell responses. So we're talking about antigen-specific responses that are unique for antigens present on the fungus. And in terms of the CD4 T-cell responses, which are very important for control of most fungal infections, Th1 responses, particularly responses that lead to interferon gamma production, and Th17 responses leading to the IL-17, IL-22 family of cytokines through the transcription factors raw gamma T and STAT3. Those seem to be the most important responses, and we know that for reasons I'll get into in the next few slides. So both primary and acquired immune deficiencies can predispose to fungal infections. The traditional teaching that we learn in medical school, graduate school, is that if you have a child with a serious infection, you think about, single gene inborn errors of immunity. That actually is probably overly simplified, because we're now seeing patients, let's say CARD9 patients, that are actually, a lot of those patients get Aspergillus infections as young adults. Some of them do. And then, of course, adults with serious fungal infections generally have acquired immune deficiencies. And the type of immune deficiency, of course, is predictive of the type of fungal infection. This is something that clinicians, don't really even think about all that much because it's so obvious. But CD4 T cell deficiencies, for instance, AIDS, that predisposes to infections with fungi such as Cryptococcus, pneumocystosis, mucocutaneous candidiasis, whereas you get phagocyte dysfunction, either quantitative or qualitative disorders, such as you give chemotherapy, the patients become neutropenic, then they become predisposed to getting disseminated aspergillosis, invasive candidiasis. Sorry, I'm losing my voice here. And there's a long list of primary immunodeficiencies that predispose to fungal infections. Back when I was training as a fellow, basically CGD was the main one that was known. So you had somebody with a fungal infection, especially a child, you would check them for CGD. If they didn't have CGD, you would just sort of scratch your head and say, this patient has something wrong with him, but I don't know what it is. Now, a whole bunch of different pathways, the CARD9 pathway, disorders in IL-17 immunity, interferon gamma immunity, they're known to predispose to fungal infections. And a lot of this, we've gotten smart to a large extent because of the mouse studies which have identified these pathways as being important in defenses in experimental mycoses in rodents. And then we've looked for, when we see humans with infections, we look for the pathways that we know are important. And so that patient with an unexplained fungal infection might have an inborn error of immunity, especially for children, but also for adults. And it seems like every day or every other day, if you go onto PubMed, you'll see new papers that describe new associations of specific fungal infections with inborn errors from interferon pathways, CARD9, et cetera. Anti-cytokine autoantibodies, and these are autoantibodies that are functionally neutralizing. They've been increasingly described. Steve Holland at the NIH has done a lot of that work. Neutralizing autoantibodies for interferon gamma. There was a very nice paper in the New England Journal that showed this, mostly in Asia. Patients with mycobacterial infections and fungal infections, for the most part, infections for which interferon gamma are very important. Interestingly, this is seen mostly in Asia, and if you have patients of Asian descent who come to Western countries, they tend not to have the autoantibodies. So there seems to be, genetics seem to play a factor, but also, of course, environment, both nature and nurture. GM-CSF autoantibodies predisposes patients to cryptococcosis, especially with Cryptococcus gattii infections. IL-17, IL-22 pathways, mucosal candidiasis, as we've heard a little bit about already. And this pathway, this subject has been introduced very nicely by Darius and Neil, my previous speakers. Not my previous speakers, but the previous speakers. But TNF-alpha inhibitors, of course, tuberculosis is the main problem in terms of infections that are seen worldwide. But in endemic areas, what they see mostly is histoplasmosis. Other mycoses have been associated. And this shows, and work by George Deep has very nicely shown the non-redundant role of TNF-alpha in host defenses against histoplasma and in mice. IL-23 and IL-17 blockers, they're used mostly in the treatment of psoriasis. About 2 or 3% of patients treated with these antibodies get significant mucosal candidiasis, very often thrush. Why 2 or 3% and not 100%? And what's different about those 2 or 3%? Your guess is as good as mine, probably better than mine. IL-6 receptor, JAK1, 2, 1, 3 inhibitors, the ibrutinib, Darius discussed very nicely. A lot of aspergillus, and most of it is extrapulmonary aspergillus. And Darius gave some nice, I think, some nice insights into why that might be. But the important point here is that we're using more and more of these drugs. I'm an infectious disease physician. Our colleagues, our rheumatology colleagues, our hematology colleagues are using all these immunosuppressive medicines. And for the most part, we're very happy with these medicines because they're somewhat selective poisons, but they're still poisons. They still inhibit a piece of the immune system. And very often we're learning, our patients are teaching us what aspects of the immune system are important in host defenses against fungi, because we treat our patients with these medicines, and then they come down with these unusual fungal infections. And finally, the last slide I'm going to leave you is a concept that I love. It comes from Arturo Casadevall and Lisa-Ann Porofsky. It's called the Damage Response Framework. And basically, it says that the inflammatory response is a double-edged sword. So too little, so here's your patient with AIDS, or here's your patient who's just gotten chemotherapy, myeloablative chemotherapy in preparation for a stem cell transplant. This patient has little to no innate and adaptive immune response. It's been wiped out. And this patient is predisposed to infections, and you may not even notice the infection until the infection is fairly far advanced, because the inflammatory response isn't doing much damage, but there isn't an inflammatory response. What's doing the damage is eventually the fungal burden becomes so huge that it starts causing problems. On the other end here, you can see death from too strong an inflammatory response. So our patient with AIDS, who is now given antiretroviral drugs, and suddenly the cryptococcus, which the immune system wasn't seeing, the immune system goes, holy four-letter word, we got to do something here, and it overreacts and trying to control it. Or our patient, our hem-onc patient, who maybe has some fungus in the lung or some bacteria in the lung, maybe a little bit of a fever, but not much in the way of pulmonary infiltrates or anything else. And then the neutrophils start coming back, and then you start seeing the pneumonia develop in the patient. So most of us live in this happy zone over here, where there's a balanced response. We're attacked by fungi, but we're able to control it without too much of an inflammatory response. And it's only when the immune system is perturbed, and that's mostly because we live in an age of immunocompromise, that we start seeing fungal infections. So I'll leave you with this, and this basically says that in the end, the fungi win. This is an artist, Johanna Martinson, and what she did was she made a city made out of bread, and with time-lapse photography, she showed how the fungi eventually destroyed the city. I think for the most part, fungi are happy to wait for us to die and then attack and grow. But when we have immunocompromise, they'll start attacking early and destroying us in life. So thank you very much.